others who might not have been able to get on the call. I also want to set up a couple of group norms because there's going to be so many people on this call. I mean, we are almost at 100 people already, and I want to make sure that we can, you know, accommodate everybody as much as we can. We'll try very hard to make sure that we can answer questions as they come. And so I also want to say that we are scientific researchers. We are not medical physicians, and so our backgrounds are that of scientific research, and we are not going to be giving medical advice. This is explicitly for the purposes of education. We are probably, with the amount of people that are here, I think it would be best if everybody puts their questions into the chat box. So anybody who doesn't know Zoom, if you just sort of go to the bottom, there's a little chat cloud button. Feel free to pop them in there, and we'll do our best to get those answered. We are also Zooming in from our homes. My home is full of people and children, so if you hear things like dishes clamoring, or my dog jumps on me, or my kids come out asking me where breakfast is, I will do my best to continue on, but I might have to pause for a second to just run our life, because we are also running our life as we work. And lastly, we ask that everybody recognize that we are all here together. We want to respect one another, try not to talk over one another if there is the opportunity to actually ask questions through your video chat. And yeah, we just want to do our best to make sure we can provide you with the amount of information that could be valuable for you. We're going to give you some background about viruses and then pop into coronavirus specifically. But before we do that, I think it's time for us to introduce who we are. So my name is Jeanne Garbarino. I am a scientist and director of rockedu Science Outreach at the Rockefeller University in New York City. This is a biomedical research facility that does some of the top science in biomedical research in the world. We are lucky to have access to amazing scientists and amazing information. And I'm joined by one of those scientists today, and I'll introduce her in a second, but I am really excited. rockedu is a program that we started in 2012 that essentially connects biomedical research communities with K through 12 for the most part, but we're extending that now because, geez, it's actually our profession to communicate science. And I will say that being on these things have made me acutely aware of chin hair, so I have plucked them for you. All those who have beards, good. I'm glad that you don't have to worry about this stuff, but you know, premenopausal woman like myself, yeah, are coming up. So no worries on the sharing, but I'm going to kick it over to Gaby and allow Gaby to introduce herself, and then we're going to jump right into our little virus education session.
All right. Well, hey everyone, just to introduce myself, my name's Gaby Paniccia. I am currently a first-year graduate student at the Rockefeller University, but I've been studying viruses for about 4 years now.
Great. So we're going to give you a little bit of a primer. I'm going to start off by talking about viruses in general, because I think it's important that we understand a little bit of context before we start jumping into the COVID virus, right? And so viruses are actually very, very small molecular systems that require a host organism to function. So something else, it requires to be on something else to function. Viruses are actually not considered to be alive, and that's because they can't actually perform all of the metabolic activities that a host cell would normally do, right? And so I want to also demonstrate the size and scale of viruses, and we're going to do that by first looking at something for which we are all familiar. And I'm holding in my hand, and it's kind of hard to see, but this is a coffee bean. Everybody should be familiar with a coffee bean, right? And I have to say, I was willing to take a single coffee bean out of my little stash for you all, but only one, because I care about you a lot, but not enough to ditch my stash of coffee. But I want to share with you my screen so I can kind of show you the scale of the viruses. And so we're going to move over to this wonderful resource that I really enjoy from the Learn Genetics department of the University of Utah. And here you can see a coffee bean, and this is Times regular 12-point font that you might see in some of the newspapers. And we're just going to zoom in and show you where we are in terms of size and scale. We see the size of a skin cell, we see the size of a red blood cell, some X chromosomes. We know what bacteria, everybody's probably heard of bacteria. Here we are getting into the bacteria sizes. And now here we are at the virus, right? Viruses are actually really, really, really small. And so we have an example of the measles virus and the hepatitis virus, the rhinovirus, and HIV, and this other type of virus called a phage. Okay. So that's a little bit of background on viruses, and everybody has, or at least the size of viruses, and everybody knows what a pencil is, right? And so if you look at a pencil and you sharpen the pencil lead and you look at the tip of the pencil, viruses are so small that 500 million to 1 billion individual virus particles can actually fit on the very tip of your pencil. I mean, that is just wild. I can't even fathom this. This feels unimaginable to me. All right, so the common feature of all viruses is that they include a nucleic acid genome. A nucleic acid genome is essentially the instructions for how to build more virus particles. And this nucleic acid genome is surrounded by an armor of proteins. Some viruses, like the coronavirus, also includes another surrounding layer, which is a membrane of fats. And it is because of this membrane of fat that scientists and clinicians are urging people to constantly wash their hands. And that is because if you think about what fats are made of, right? So fats are these molecules, and they're very common in all sorts of things that we do every day. If everybody's cooked something like a grilled cheese sandwich or some pork chops or something in a pan, you can't, and there's all that grease layer all over your pan, you can't necessarily take that pan and just put it under plain water to clean it, right? You have to add soap. And that is basically the same physical principles of getting the virus off of your hands. Is you need that soap is because of that round membrane layer. And so a virus exists to perform a single overall function, and that is to transmit copies of their genomic instructions into a host cell. And they want to get that host cell to make more copies of the building blocks that can create the virus. And then they want the host cell to help them assemble those pieces to make new virus particles, and then release those new virus particles into something that they can continue to infect. So they want to go from one infected cell and then make a bunch of stuff and to go into more infected cells. And as I had mentioned, they need to infect these cells because viruses do not themselves possess the ability to actually read their genetic instructions and assemble more copies of themselves. So they hijack the machines inside of host cells that normally do this, and they get rich off of the resources and work of others. All right, so where did viruses come from? While it's really hard to study how viruses emerged on the planet, especially since viruses are really small and hard to detect, and we can't really look at like a fossil record. I mean, while we can consider their nucleic acids as a kind of a fossil record, but it's not like we can physically look at rocks and find like bones or anything, because viruses don't have those, right? And then they also emerged a really long time ago at a time when the Earth looks extremely different than it looks right now. And so there's some evidence that suggests that viruses could have emerged even before the first cells on this planet. And then there's another theory that suggests that viruses evolved along with cells and represent escaped genes from these cells. And so genetically, viruses tend to look a lot like the cells that they infect. And this is a really important thing when we think about like how viruses jump from species to species. And some of them have even kidnapped host genes into their own genomes to help them better infect cells. They're incredible molecular machines. But whichever camp folks are in, there's no denying that viruses are really, really old. And because they've been around for so long, it's no surprise that there are millions of different types of viruses that infect all cell types. And we're talking plants and animals, and there's even viruses that infect bacteria. And the majority of viruses do not actually impact humans. There are a ton of different viruses on us and in us all of the time. Most of these actually infect bacteria. And in fact, 40% of all bacteria on Earth are destroyed each day by the special viruses that infect them called phages. Of course, they keep growing. The bacteria keep growing faster than the phages can kill them, which is why we still have bacteria. And so when we think about how many viruses are around us, one tiny drop of seawater could contain like 10 million virus particles. And so as a whole, viruses are, as like a whole thing, are not really that scary for humans. However, there are some viruses that do impact animals and plants, and we are actually an example of animals. And so we are currently in a situation where humans are facing a brand new strain of virus on a really big scale, and we're learning new information about it every single day, if not every single hour. And so here we're hoping to give you some of the information that we currently know about COVID-19 and provide an opportunity for you all to ask questions. And I'm going to kick it over to Gaby because she is really smart and knows a lot more about coronaviruses than I do. So I'm gonna let her take it from here.
Okay, cool. So coronaviruses are actually a really large family of viruses, and they're called coronaviruses because they're named for the sort of spiky crown that surrounds the virus. And this crown actually serves a purpose. These are all of the receptors that they use to bind to host cells and essentially trick the host cell into getting infected. So inside of a coronavirus, like a lot of other viruses, it has its genetic information stored in a single strand of RNA. And RNA is basically like DNA, it's only a little bit different chemically. So functionally in a cell, DNA has to be translated into RNA before it can really do anything. And so the virus, by being already RNA, is sort of the minute it gets into the cell, is sort of ready to go. You've also probably heard a lot of names for this coronavirus. COVID-19, SARS-CoV-2, the novel coronavirus. All of these names, despite it sort of just making it sound like, Game of Thrones Khaleesi level of just nonsense, they all actually do have a meaning, and they're naming it for different facets of what it is. So because this one is new, that's why we called it the new one. Co comes from coronavirus, VI from virus, D for disease, and 19 for the year that it appeared. And like I said, there are a lot of different species of coronavirus. So this is not necessarily like, for the most part, we've seen these viruses before, the ones that cause the common cold. So it's not like every coronavirus is, the end of the world, because we only really hear about them, I feel like, in the context of SARS previously, MERS previously, and now this. So I'm actually going to, I should be able to screen share, I think, right, Jeanne? Walk you guys through, there's a really great New York Times article that goes through how the coronavirus gets into your cells. And so I think it's a good infographic for me to walk everyone through. Let's see. This is the first time I've actually done this. All right, can everyone see that? Is everyone looking at what I'm looking at? Okay, cool. So essentially, this is the form of the coronavirus. I'll scroll up a little bit. This is what it looks like from the outside. So these spikes, those are what I was talking about that gets at the name, the crown. And those are what bind to cells. And then if this is a cross section right now that you're looking at, so in the inside is coiled that single strand of RNA. That's its genetic material. It's got the spikes sticking out through it. And then this layer that's in yellow is a protein and lipid layer. So as Jeanne was saying, the fats are actually really important in the process of it getting into cells, and you'll see that in a second. So the outer part of the spike binds to a receptor that exists in our airway cells. So this is why it's a respiratory virus and why it's not bloodborne, because these receptors, that's essentially the lock that it fits its key into, they don't really exist in other parts of the body, or at least not in enough numbers for you to get much of an infection rate. So once it reaches a vulnerable cell, the cell basically eats it. I don't know if it's shown really well, but you can see that this is curving in here, and it puts it in this bubble. So once it gets into this bubble, the virus has the next problem, which then it has to escape from that. And that's where this membrane made of fats comes in, because the bubble that the cell puts it in is also made of fats. So it sort of fuses with that to then kick its RNA out into the cell. And then once that's released, then it starts using the cell's machines to take it over and use and essentially make more virus. So it gets the viral RNA translated, is what we say. It's essentially when the cell reads the blueprint that the virus gave it and starts making virus proteins. So the virus is actually pretty small. So essentially, once your cells have been converted to little virus factories, new viruses get assembled and then pop out. And that's what makes you infectious, essentially. So I'm going to stop screen sharing. Let's see. Everything should have gone back to normal.
Yeah, we're back to.
Awesome.
Cool.
Hopefully that little explanation made sense for everyone. Apologies if it didn't. Scientists can sort of assemble an idea of where a virus came from based on something called codon optimization. So in the genetic code, different animal species spell things kind of differently. They use different letters at different times. Think of it a little bit as like American versus European spelling. We spell color C-O-L-O-R. Europeans might spell it C-O-L-O-U-R. It doesn't make a difference for how it's read. It just makes a difference for how it's spelled. Same thing for viruses and for different animal species. So what we can do is, because viruses have to match the spelling of their hosts so they make sense, we can look at that and sort of compare it to what other animal species it's similar to. And so because of that, we were able to sort of figure out it probably came from bats. So I guess another question is, why did this spread so quickly? And this was a really like all of the stars align sort of situation. A virus jumping species is extremely random. It's just random, tiny genetic variations that probably meant nothing to the bat virus's ability to be a bat virus. But then all of a sudden, when it was exposed to people, it let it infect people. If this had happened somewhere with less people, we might not be in this situation right now. So for example, if this had jumped from penguins in Antarctica, all of, I think, 20 people in Antarctica probably wouldn't have been able to spread this to the entire world. They would have just gotten sick and still been cold. But there's also other factors that I think really played a role that's unique to this virus. So long incubation time and asymptomatic people. So the long incubation time essentially means that it can take up to 5 days or longer for symptoms to begin, but you can be infectious before then. So this means that people could have gone about their day and infected their friends, family, and coworkers without even really knowing that they were sick, or that they were shedding virus, basically. And having asymptomatic people essentially contributes to this too. So it seems like a lot of people around my age, between 20 and 30, may not ever show symptoms of having the virus, which essentially means that people went around their whole day and accidentally got other people sick without ever knowing that they themselves had the virus. So that's a big part of why, I'm currently social distancing. Even though I'm not sick, I'm hanging out just in my apartment so that if for some reason I am sick, I don't get other people sick.
So I think maybe we can talk a little bit more about why we're social distancing, because I mean, it feels a little silly to just say, let's shut down life as we know it and all remain in our homes, right? Like, especially if we're seeing people, like not a lot of people are really showing symptoms. Like Gaby just mentioned, people in her age group are not necessarily showing symptoms. And we know that kids, for the most part, are not showing symptoms at all. So if there are so many people who are not showing symptoms or experiencing a mild kind of illness in response to the virus, like why would we social distance in the first place? And the answers to that are actually pretty interesting and complex. And because the virus is very new, like humans overall don't have our own immune system, our own innate immunity for that right now. We've never had to remember this in our immune systems in the past. We also know that it is spreading very quickly, and that's mostly because we are a global society. We are on planes and trains and cars, and we mix in large groups all the time. And because there are so many potentials for asymptomatic transmission, I mean, things can go really fast, right? And as I had mentioned, a lot of people are not getting sick from this, and those who are for the most part, are actually mild. But here's where the social distancing is really important, right? There are specific vulnerable populations, like populations who are older, populations of people who have other types of illnesses, and people who have behaviors that could be associated with health problems like smoking, right? And so there are enough people in this category for a specific town or city or region that if all of the vulnerable members of that community became sick at once, there wouldn't be enough hospital beds and doctors to take care of them. And this essentially is really, really a big deal for our healthcare systems. It would mean our healthcare systems become overloaded, and the normal level of care that we would see, on when we were able to hang out physically and not virtually, we would see that this care level would be significantly decreased. And if this happens, people who get COVID-19 may not be able to get the best care that they need because doctors and nurses and other hospital staff would be stretched really thin. And we've seen this thing happen in other places where they were not able to adequately control the spread, such as Italy. And it also means that not normal medical care or even non-COVID-19 emergency care would be significantly slowed. And this is not good, right? So if anybody becomes hurt, or, somebody has a heart attack, or, some other kind of medical emergency takes place, I mean, it would impact our ability to respond to that too, right? And so even though it feels a little silly, staying home means that we're actually protecting both ourselves and protecting each other, protecting the people that we love. I would say it's impossible at this point to consider that I think at this point, somebody we know probably has the coronavirus at this point in time. And we just don't know, they might be asymptomatic. So let's just minimize how much we're interacting with each other. It's also a good reminder that we are a species, right? And in order to survive, sometimes we really need to come together. And maybe this could be a lesson for us for some of the other things that we need to address as a society, like climate change, but we can talk about that another time. And Gaby, do you want to talk a little bit about virus extinction?
Yeah, I was gonna say, I wanted to jump in there really quick.
Yeah.
So there's something really interesting about virus extinction. So SARS is extinct, the first SARS. And it's something that we don't have to worry about getting again because we made it so that it was not spreading quietly throughout the population. And I think what part of the push is, to keep us all, social distancing and prevent it from spreading is we don't want this to become the next common flu. I mean, you hear the common flu cited all the time as being more deadly. That's just because it infects more people so often. And we don't really want to be fighting this virus forever. We don't want to have to get a new, coronavirus vaccine shot every year. Although, based on some structural differences, we may not have to. It really depends. The flu is particularly unique in the way that it changes year to year. But we really want to avoid that. We don't want, like I said, we don't want to have to be fighting this virus forever. So I think our goal right now is to really quash it as fast as possible to make sure that this isn't a recurring health problem.
Yeah. So should we get into where we are with the cure, Gaby? Do you want to talk about that a little bit?
Yeah, I can definitely talk about that. So there's pretty good news. Is there a cure? Your own immune system.
No.
So most people only have mild symptoms and recover completely. Your own immune system is really good at fighting these things off. And sorry, 1 second. Afterwards, after you fight something like this off, your body's pretty good at remembering that it saw it in the first place and fighting it off again. We are also, as scientists, working a lot on ways to stop this. So there's 2 different arms that scientists go about preventing, or it's treatment and prevention. Those are the ways that we approach viruses. So treatment, we tackle those through antivirals for the most part. So antivirals aren't antibiotics per se. They're things that really slow the progression of the virus. So I think right now the two that I think people are like specifically for this more so are things called nucleoside analogs, where essentially they're like messed up letters of the genetic code. And the virus can't really spell too good, it can't really tell that those are different. And so it winds up incorporating those in its instructions for the next virus. And so the next virus, when it gets those messed up instructions, can't execute its functions properly because all of the instructions are scrambled, spelled backwards, stuff like that. And so that's essentially the purpose of a nucleoside analog. And I think there's two that are currently being tested right now, predominantly in places with higher density infections like China. So they're a little ahead of the curve on developing that. There's also, for the prevention angle, which is vaccines. Vaccines take generally a lot longer than antivirals because it's hard to make sure that you're going to have protection forever, basically. So for example, think about the shots that you probably only got once or twice as a child and have immunity now. It's kind of hard to make a vaccine that perfect. So it requires a lot of refinement. But at the same time, what we're not looking for is perfect. Right now, the vaccines that we're probably going to be looking for are things that have some small reduction. So essentially something that we can give healthcare workers to reduce the likelihood that they'll get sick. And so a lot of these things right now are really going to go into treating the people who have bad cases, where the antivirals would probably decrease how bad, like decrease their symptoms and reduce how bad their symptom load is, and also preventing healthcare workers from getting it, and help them stay on their feet for long enough to keep, the people who are the most sick healthy.
So I think we'll start pulling the questions from the chat box. And we have a question from Laura that asks about how long the viruses live on surfaces. And this is a really good question, and there has been a lot of work on this in the last couple of days. And there was a recent paper that was published just the other day that that says, hold on, let me find all of my data so I don't misspeak. Oh, the stability of the virus is definitely dependent on the surface. And for example, plastic and stainless steel has showed the presence of viable virus at 72 hours after being exposed, whereas a material like copper or cardboard only doesn't show viable virus by 24 hours. And I think this is like the whole material thing is a really big thing that we have to consider. And this is why people are suggesting that what if you live in a building, you're wiping down your elevator buttons on a regular basis, you're wiping down your doorknobs. And it's probably why a lot of places are deciding to close their playgrounds. Even though we want to get out and we want our kids to burn off some energy, a playground is mostly made of plastic, right? And if we can consider kids as little vectors that are, it's not really dangerous to them, but they can spread it to all these other people, it's really important that we're taking these measures to make sure that we can maintain safety for everybody. And so in terms of how long it can transmit from a specific surface, it depends on the surface, and more data is probably going to come out on that with more specific guidelines in the coming days or weeks, I would say. Okay, what other questions? Let's see what we can answer. All right, hold on. Sorry, there's a lot of...
yeah, we have a lot of questions.
Yeah, I'm trying to go through them
as fast as possible in the chat,
yeah.
if you don't think we can get to them. So I'm trying to think. So I want to answer one that I saw real quick.
Yeah.
Coronavirus is not a retrovirus. You do not have to worry about having this forever. It is a transient infection. So that just means that, like a cold or a flu, once it's done, it's done. You are not infected with it. But part of the reason why we're still worried about coronavirus is because it seems like people might shed virus, essentially are no longer sick, but still are in the process of clearing that virus and may still be infectious to others. So it's probably safe to treat, even if, say, you've been in quarantine for 14 days, you've been sort of on lockdown in your house, and you think that you're better now, to stay inside for an extra couple of days to make sure that you're not, that you're fine. And I don't know if we're doing secondary testing for people who think they've recovered yet, because I know the US
There's
has?
some, I think, very small data sets coming out of places in China and South Korea. But, you know, they did just come out with a paper this morning, or last night actually, on bioRxiv, which is a repository for people to put their reports in before they become peer reviewed. So it's a really, really amazing resource for scientists because it allows for information to be pushed out really quickly. But they did, I'm not sure if you just mentioned this, but they did show that in monkeys who had been infected with COVID-19, they're not getting re-sick, they're not getting reinfected, which suggests that maybe our immune systems, you know, it's sort of like a one and done. Once we get infected, it's quite possible that we're not going to get reinfected. And so I think that's a really positive finding for people. There is a question. Oh yeah, go ahead.
Well, no, I always see something about like people asking a bunch of questions about when something mutates.
Oh,
And I always want to tackle this because it's a really interesting thing from a science perspective that a lot of people who aren't scientists aren't exposed to. So mutation sounds scary. Everyone thinks of, you know, what, like Contagion or something like that, like some crazy sci-fi Andromeda strain kind of thing. It's not what it means for scientists. So viruses mutate all the time. All of us mutate all the time, but it's not usually good. So for us, we're mutating constantly. That's how eventually some of us get cancer. But our bodies are pretty good at cleaning it up. The virus mutates too, as the goes from, you know, inside your body and from person to person. But usually this is way more likely to produce non-functional virus or virus that's worse at being a virus than some magic superbug. Mm-hmm. And I want to explain a sort of concept in virology that I think is kind of important to refocus how we think about viruses. A virus's goal is not to get you sick. Its goal is to spread. If making you sick helps with that, so for example, making you cough onto other people so you spread it, the virus will probably always have coughing symptoms. But for the most part, the viruses that I think are the best viruses at being viruses are the ones that don't make you sick. Or sick. Think about the common cold. Everyone gives each other the common cold, and we don't really care about it that much. Same thing about, there's a virus that I think 98% of us have, or at least have been in contact with. It's called Epstein-Barr. And none of us get sick, or a small fraction causes mono, but that's like a very small fraction considering how many people are exposed to it. So the concept of the virus mutating doesn't mean that we're going to get a super mega virus. What it means instead is something a little bit closer to why we have to get a flu shot every year. So we're going to probably, there's sort of host and virus tug of war that happens. And we talk about virus-host interaction as scientists and evolution as scientists. And so if anything, we're worried the most about the outside of the virus changing a little bit over time, in the same way that the outside of the flu changes over time, where essentially we're going to need a different or multiple vaccines. And giving it the least amount of opportunity to change is part of why we're trying to take all these measures now. So that, like I said, we don't have to get a shot for this every year, or we don't have to develop, you know, multiple stage vaccines because it looks a little bit different on the outside. And the look of that virus is essentially how our bodies recognize it to begin with.
Great. Let's see what we have for more questions. We have somebody on here, Luke, who is also answering some questions for us. So thank you. So check out Luke's answers in the chat.
I was gonna say, Luke, just pop in the chat. Like, I'm just curious. Like, what do you do? Like,
yeah.
Are you from Rockefeller? Like, it's cool. Yeah. Like, I mean, we have other scientists who are in the chat too. So I'm just kind of curious.
Okay, so we saw that coronavirus is not a retrovirus. There were some other questions that maybe we can talk about, including maybe how we test for SARS-CoV-2 virus, right? And so as Gaby mentioned, the SARS Cov-2 virus has a set of instructions inside of it for building more copies of itself. And this particular virus uses RNA to write these instructions. And RNA is a type of nucleic acid, and it has a lot of similarities to another molecule that we're very familiar with. I think most of us are probably familiar with DNA. So our bodies have both DNA and RNA, but in this case for the virus, the RNA is the main source of genetic instructions. And just like DNA, RNA has 4 letters, and it is the sequence of these letters that sort of form the recipe for building the virus. And in order to test for COVID-19, scientists and clinicians have to extract RNA from a sample. And usually this sample is, you put it like one of those nasal, like Q-tips, those really long Q-tips up your nose. And then you use all the, I'm just going to use a very scientific term. You'll use all that goop to get the RNA out of that nasal swab. And then you make a ton of copies of that RNA so that there's enough of those letters that we can read it. And then we look at the sequence of those letters to see if those letters match to the COVID virus or the SARS-CoV-2 virus. And so that's how the virus is tested, and it takes a little while for that to happen. It's quite a process because you have to do the RNA extraction, which is a several-hour thing, and then you have to make a bunch of copies, which takes a couple of hours, and then you have to sort of read the letters in those copies, and that takes a couple of hours. And so it's a long process. It's not like when we go to the doctor and we take one of those rapid flu tests where we know like fairly soon. So right now it's a very slow process, and it is a bottleneck for community testing. And I think we're now ramping up our capacity to do this on a larger scale, but until now it was very specific in terms of who was eligible to get that test.
I also really quick want to mention something because it ties in with testing. So a bunch of people in the chat are saying that it's spreading lightning fast. It's not really spread that's happening right now. You're watching cases go up dramatically in the last couple of days, especially in New York. This is not because in one day it infected, 1,000 more people. It just means that we tested way more people. So in reality, part of the reason why everything's shutting down is because we expect that our estimation based on confirmed cases is much lower than the amount of people who actually have come in contact with the virus. So it doesn't mean that like, ooh, you may already be infected. Like, I don't want to like scare you guys like that. But it's sometimes scarier when you think that it's spreading lightning fast, that like in a day we're all going to be diagnosed positive for coronavirus. In reality, we just really lagged on testing. And so we're trying to catch up with that now. It's essentially like, was it China or South Korea that started when they bumped up their testing? They had a huge spike.
Yeah, I think South Korea.
I think it was South Korea, yeah.
Massive community testing, like everybody's tested. Yeah.
So what happened was if you look at their statistics, there's this huge jump that makes it seem like in a day, everyone got it. But that's not accurate. It just meant that over the last, 3 days, that many more people had got it and they suddenly were now able to test for it. So it's gonna seem scary over the next, week as the US really ramps up its testing infrastructure, because it's gonna seem like cases are popping up out of everywhere, but in reality, they were already there.
Yeah.
Which is also worrying, but that's why we're social distancing now before, we reach some threshold number of cases.
Yeah, and again, just to emphasize, while more people probably have the virus than we can detect at the moment, again, it's the majority of people will either be asymptomatic or have mild symptoms. It's really the vulnerable populations that we are concerned about, and the overwhelming of our healthcare systems. And so I think for the most part, people should be vigilant. They should practice the important things that we can do to protect each other, like staying inside, having 6 feet of space between us if we do end up going out, washing our hands, trying to avoid touching our faces, and like wiping down things like, doorknobs and phones and keyboards and things that other people can touch. And so, it feels a little scary to be in these situations, but I think overall, there are people out there worrying about this for you, and, just do your part and, snuggle with your pets a little bit more, maybe up your watching of Netflix or Hulu, so I think, we can sort of ease some of the anxieties that we might have, because people are on this. And I also want to emphasize the wonderful, wonderful resource that the scientists and clinicians in Asia have been providing the world. The work coming out of China, the work coming out of South Korea, I mean, we cannot be more grateful for the wonderful information and the resource sharing that they are providing to our communities. And so I really want to emphasize the value of the work that these scientists have gone through. And, you know, being on the front lines, I mean, they were the ones who helped warn the world that this was coming their way. And we really should be grateful to these scientists and physicians for their service.
I'm seeing some great questions that I want to answer.
Yeah.
So two things, it seems to be about prevention and also testing. One, that's, you told that testing kits are limited. What exactly are we lacking in? So there seems to be a couple of things. Then each testing kit might be a little bit different, by the way, just based on what bait they use to fish out the genes of the virus. We call those probes. And what it seems like we're limited in right now, or at least what I was reading earlier, is the kits that we use to extract the genetic information from the virus. So RNA is notoriously less stable than DNA. It degrades kind of fast, and you want to make sure it doesn't degrade so that you can read it properly. And so that is, you need really, really clean materials, because there's a lot of stuff all over everything that we touch that degrades RNA. It's just sort of in the environment. And so we need stuff that will stabilize it and will prevent it from being degraded. And that has to be made really carefully. A lot of us as scientists can't make that in our own labs because we don't have, you know, a ton of chemicals and the space for it. So we rely on a lot of the places that make these kits, these RNA stabilization kits. That's essentially like use buffer 1, use buffer 2, use buffer 3. It's very like straightforward. And that's what's limited. It's not necessarily that there's a limited amount of scientists, because the process is actually very easy. Even me as a first-year graduate student, I could have done this as an undergrad. But we are actually limited in clinical lab space, people who are certified to work with patient samples. Like I'm only certified to work with like stem cells. Nothing that's come like really, really directly out of a person. And then someone else asked about masks. So part of the thing about masks is that they're going to be in short supply. For healthcare workers especially, it's a lot more important for them to wear masks to be able to have some sort of barrier in case, you know, the people who are sick who they're working with sneeze on them. For us just going to get our groceries, you don't need a mask, and you don't need to be hoarding masks to take them away from people who need them. Masks also, to speak a little discouragingly for you guys, the masks that we use in a BSL-3 facility where we work with dangerous viruses, those are the N95 masks. People have been snagging them up so fast that, one, researchers like myself who need them can't get them. And two, these masks you have to be fitted for, and they have to be really, really specific. And so I've seen people out on the street wearing N95s with big gaping holes in the side of them that do nothing to help you. So using masks for trained healthcare workers that you yourself don't know how to use is not going to help you. Similar thing with surgical masks. They're not like the end-all be-all, you're now in a bubble just because you have a mask on your face. And in fact, a lot of people wear them for so long that they get wet and stuff can still come out, essentially. So wear one if you're sick, because it is a good barrier. You know, if you're coughing, then stuff's not going to fly through that as much. But if you're just a regular person, don't be hoarding these masks, because the people who are really needing them really need them.
Yeah, and if I can add just one thing on the RNA kits, there is a company called QIAGEN that is drastically upping the production of these kits so that they can be distributed to scientists and clinicians who are doing COVID testing. And then secondly, regarding the N95 mask, I think Gabby makes an excellent point. in that, you know, being fitted for this is essential. And one of the processes, just to explain what that looks like, somebody who would be fitted for an N95 mask, what they would do is they would put on the mask and they would go into a small chamber, and then there would be some aspartame, which is, you know, people have tasted like an artificial sweetener. Aspartame would be aerosolized. And if you can taste it, then you have to get back out and then get the mask refitted. And it's a series of getting your mask fitted and then spraying aspartame to see if you can taste it, and then going back out several times. It takes healthcare workers sometimes several times to get this right. So to consider that we, just regular people, could just go pop on an N95 mask and be protected is actually not a real thing. And so please, everybody, do not purchase N95 masks just for your home use. You are taking valuable resources away from healthcare workers. And this is a really important product that we need to make sure that they are safe, so that we end up still having doctors and nurses who can help us.
I think also, I was going to say, I'm seeing other questions about ibuprofen, because this is a thing that happened recently. So the CDC, I think, or the WHO put out a thing about you should probably avoid ibuprofen if you have coronavirus. So this seems to be an interesting thing. I've been looking into it. I don't think, it means that, you're going to die if you took an ibuprofen and you're sick. What it seems like is happening is, so there's some older data from a while ago. I don't think it used coronavirus. I think it was about the flu, that it didn't seem to help as much. Those are different viruses. So I think currently it's based on a lot of anecdotal things from nurses and doctors that they're seeing people who are taking ibuprofen might be coming in with worse symptoms. Because there's no statistics yet, I am a little leery of how pronounced this thing is. Essentially, it's a little bit of a thing about how if someone tells you to look for something, you start seeing it everywhere, you learn a new word, so all of a sudden you start hearing it. I don't know how much of this is enough people finally, like a nurse asked like the first 10 patients who walked through the door with it, did you take ibuprofen? They all took ibuprofen, and maybe 2 of them had more advanced symptoms. So then that nurse thought, okay, now it's ibuprofen that's doing this. I don't know if it's correlation as opposed to causation, is something that you'll hear a lot of scientists say, but I would keep looking stuff up as this continue. I would trust the WHO, which essentially means, if you have ibuprofen, use it for a headache. If you start coming down with coronavirus and you have Tylenol, use that instead. That's basically my standpoint on this.
So there's a couple of questions around the anticipation of how long we need to maintain social distancing. And so I want to try to address that. And I just want to say it's very hard to predict exactly how much time we're going to need to do this, right? It's clear that we have to protect each other, and it's clear that business as usual, or at least sort of how we thought about normal life, definitely needs to shift. We have to continue to assess how well our measures for social distancing are working, or maybe not working. We have to assess where we are on treatments, including how efficiently we can distribute these treatments to those who are facing any kind of COVID-19 issues. And we also have to consider how long it will take us to get an effective vaccine. Not to mention also developing our own innate immune systems to be responsive to any of this virus. There's some good news in a recent report that suggests that reinfection is unlikely, meaning that there's evidence to suggest that once you get this particular strain, you become immune to it, as we sort of talked about before. But we do need more data on this to be entirely sure, and at least the work is promising. And I want to, while we're facing these uncertain times, it's actually not entirely clear. Some scientists are saying that we have to maintain social distancing for actually a really long time, maybe something like a year, but I don't actually know if that's something that we can say with any kind of confidence without some of the more information that we're getting regarding these other things that I just discussed. But there is a quote from Dr. Fauci, who is basically leading the efforts in our government. I think they are very, very respected, and I think he has a really good take on things. And the quote that he says is, and the thing I want to re-emphasize is, when you're dealing with an emerging infectious disease outbreak, you are always behind where you think you are if you think that today reflects where you really are, right? And so it's really just getting a handle of what's happening. We're still getting data, we're still processing the data, trying to understand what the data mean. And I think what we all have to do is just be patient and listen to trusted sources and think about how we can just work our lives into this new reality of social distancing for now. I mean, I don't know, whoever bought stock in Zoom, you are now rich.
I also noticed, so I saw someone post a question a while ago before it got drowned out by chat nonsense, about quinine and chloroquinine. I never know if I'm pronouncing that right. That's been an interesting thing that's come out of this. Someone asked about the chemistry of it. My PI actually sent us, PI, the head of my lab, he actually sent us a whole big thing about the stuff that's been coming out about this. According to what they were saying, hydroxychloroquine or chloroquine, they are used predominantly against malaria, which is a little bit wild that someone thought to use this in the first place. It seems like it raises the pH, which is essentially low pH is acid, high pH is base. And of that little bubble that the virus gets sucked into when it gets put in the cell, the virus relies on it being acidic. That's how it knows it's inside a cell, and that's how it starts pulling itself close to that bubble so that it can dump its RNA out into the cell. And so what this does is it seems like it might slow the stages of early infection, and help like contain it within a person a little bit, by making that bubble a little bit less, cushy for the virus. Not giving it as easy of a time to push its RNA out into the cell. So it's pretty interesting. I think there are clinical trials in China, South Korea, Japan, France, and the US. So I feel like we will probably see this used for healthcare workers or those at high risk of infection. So someone was talking about in the chat cystic fibrosis. That would probably be a person that would be able to get this if it starts rolling out. These are early tests though. I don't think they've been done on a lot of people. I think their phase, I'm trying to look for the phase because that tells me how far they've gone. I can't find it right now in this email. But if that starts rolling out and that seems promising through continued trials, that might be something that we start, seeing distributed in hospitals and to those who are the most at risk.
Great. So there's a ton. I feel like I have something that says 99 plus new questions. So we're trying to go through these a little bit so that we can make sure that we are doing our best. So let's see. I think there are some questions around people with immune suppression, and I think these are people who do have autoimmune diseases or immune suppressions of any kind, or taking medications to suppress their immune systems. These are the people that I think really need to work on really being very vigilant about social distancing, and ask the people around you, if you're in a home with anybody, to also make sure that they are vigilant about social distancing. So I think that's really important. Let's see, what other questions do we have going on? Let's see.
I'm answering the ones about flu tests right now. I would suggest scrolling up a little bit. I'm sorry, I'm trying to, like I said, I'm trying to get through as many in the chat as I can.
Yeah.
You might just have to make like an ongoing.
Yeah. And also we are approaching a 1 hour time point, so maybe we can try to answer 1 or 2 more questions, but I think, we should probably take this, and we'll try to post this on YouTube, and we'll also try to, provide more instruction. So rockedu is actually generating a whole resource around viruses, and you can look for that at rockedu.rockefeller.edu. Codon.rockefeller.edu. We have a bunch of teaching resources that we could use, and we also have a blog, and so we'll try to get all of this stuff on our blog. And you can also follow us on Instagram. We'll probably be doing a bunch of Instagram stories around this as well. And I am going to type in our Instagram handle so that you can see that. And I think some people on my team, so yeah, Desan is on my team, Lizzie, Odellis, Lizzie, if you can post, we have an exit survey. So if you can tell us about how this went for you so that we can continue to do these things. And so look at the chat box and please fill out our survey so that we can hear from you. How did this go? Did we hit the right levels? Is there something that we should do to make your questions a little bit more reasonable, or I don't know, like where are we, how are we doing? Let us know how that's going. Cool. I think maybe do we want to hit one more question and then maybe we'll call it a day for now?
Let's see. Let me look at some of the new ones. See if,
So I think there was the thing about the flu tests are quick and easy, and they're just a different type of test, right? And so
yeah.
they're based on a different type of measurement, and you could do that really quickly in a doctor's office. Maybe we'll get there with COVID. We just don't know what kind of markers to look for right now to other than the straight up DNA, or straight up RNA. Excuse me, it is RNA. Yes.
We are working though very fast. There's been a lot of I really think I read somewhere like Ireland that like some scientists there developed like a faster test. Like a lot of this stuff. So someone says they make a faster test. We obviously have to verify that. Scientists like data. So what they try to do then is they try to roll it out and see how it works, and they have to compare it against the tests we already have. They can't just roll out a new test and not have anything to compare it to, because then how do we know that it's not diagnosing people, right? Fortunately, at least with the test that we have, even if it takes a while, the rate of a false negative, as in saying that someone doesn't have coronavirus when they do, is really low. So if you get a coronavirus diagnosis, or you or someone you know does, you can have pretty good confidence in the science that says that you have coronavirus. Don't think, they're wrong. No. It means you have it. You should definitely take that to heart.
Yeah. Cool. All right. I think we are probably going to call it a day. Look for this on YouTube. If you had people who were not able to join, but you think this could be of value, we'll get this out. I don't want to make any huge promises, but maybe we can have it by tomorrow. Don't hate me, Lizzie, if you're helping with this process. But we're really trying to figure out like how we can be, like, again, we are part of a science outreach community, and it is our profession to connect people to research information, and we usually do this in person, and so we're really trying to figure out how can we do this in an effective way when we're not allowed to be in person anymore. And so yeah, continue being in touch. I'm gonna be a risk taker and throw my email in the chat box. People are like, don't do it, but I want to make myself available if you have ideas. We are also hoping to connect with both New York City teachers and New York City students through some other initiatives where we can maybe pair people with scientists to try to figure out ways for helping people with their education in those ways. And I am glad that everybody is giving us some pretty useful feedback. Don't forget to answer our survey. The link is in the chat. And here is my email address. If I get bombarded, I will try my best to get back to you. Okay. Well, thank you again. Be healthy, be well, wash your hands, stop picking your nose, stop rubbing your eyes. I mean, and when we're all on here, I'll continue to pluck all of my chin hairs so they're not all over the place. But whatever if we do. Okay. All right. Peace out, everybody. Enjoy.